Okay. So today we are going to talk about the first text, which is the Sompayrac text. We're going to essentially do 2 lectures on the Sompayrac text, although I've kind of chopped the reading into 3 parts, so the first few chapters hopefully weren't too tedious to get through. Excuse me. The next lecture will be about 5, 6, and 7, and then the last 3 chapters are definitely worth reading, but I'm not going to give a lecture about the next one. We'll do review questions at Jeopardy about. Okay, so I'm going to start today with a few warm-up questions. Think about them for a second and write down your answer, and we'll come back to them at the end. But what DNA segments are the first to rearrange in the adaptive immune cell? Question 2 is, which antibody is the best at fixing complement? Or if you would like, what are the first best 4 antibodies at fixing complement? And then question 3, what molecule causes the release of the clip or invariant chain in the MHC Class 2 presentation? Okay. So, first thing we're going to talk about is the innate immune system. The innate immune system we use in contrast to the adaptive immune system. So the difference is if something is innate, it doesn't change from when you're born. It's always there. As opposed to the adaptive immune system, which adapts. The DNA actually rearranges and changes in order to work even more effectively. In the innate immune system, all the proteins and cells of the innate immune system are the same no matter what. There's no changing. So that's the one of the first big breakdowns in all the terminology we use in the immune system, the innate immune system versus the adaptive immune system. Okay, so first we're going to talk about some cells and proteins in the innate immune system, the first of which are the complement proteins. The complement, you probably recall from med school, is a system of proteins that work together to kill pathogens. The complement system, I think, can, at first glance, be fairly overwhelming. But again, we're just at the beginning, just kind of take it in and start to remember that it's a series of proteins that cascade off each other to eventually punch holes in cells and kill them by creating this osmotic gradient. So the complement system can get started via 3 pathways: the alternative, the classical, or the lectin. We're actually only going to talk about the alternative and lectin today. In this part, we're just going to talk about the innate immune system. That classical, of course, is activation by antibody, and that bridges over to the adaptive. Okay, so in the alternative pathway, which I think is maybe the easiest to understand, the first step is spontaneous. In the 3 methods of starting the complement system, it's really this first step that's different. All of the rest is extremely similar or, in fact, the same, but this first step makes all the difference. So in the alternative pathway, C3 is just spontaneously cleaved into two smaller proteins, C3A and C3B. That's it. It just spontaneously happens. So we're going to forget about C3A for a minute, but C3B is the one we're going to concentrate on. And what it's good at is binding to carbohydrates. And it binds, or it knows something's a carbohydrate because it's looking for an amino or hydroxyl group on that carbohydrate. If it fails to very quickly bind to a carbohydrate, then it's neutralized by the water molecule within 60 microseconds, so very, very quickly. Now, carbohydrates are something that are fairly specific to bacteria. So this is one fail-safe the immune system has to not land on your own immune cell and start attacking it. So it's going to preferentially bind to carbohydrates. If it doesn't see a carbohydrate, it's going to be almost instantaneously neutralized. If it successfully binds, though, then something called protein B, which is just floating around in your body like C3, the C3b will then bind to protein B. And something called protein D clips that B, so then you have a complex called C3bb. Now, again, don't get hung up on all these steps. Just kind of have a sense that C3 gets clipped and then binds to a bacteria, and then B comes on board. Now, the C3bbb is a convertase, and what it does is it cleaves more C3, so you can amplify this process and dig lots of holes into the cell that it's landed on. You can also cleave C5 into C5b, and that's the first step of the, that MAC complex, that attack complex. Once C5 is turned to C5b, then it lands and it creates that MAC with C5b, C6, C7, C8, C9, and it punches a hole. And it can do that over and over and over. And that C3bbb can cleave over and over and over and over. Okay, and that's it. That's essentially the complement system. So, right. So we're going to let me step back. Right. So that's how the holes get punched. It's just a chain reaction of proteins that just act on each other to punch all these holes. Now, there are other fail-safes to make sure that your complement system isn't punching holes in your own cells when it shouldn't be. So one of these fail-safes is called the decay accelerating factor, or DAF. Only found on human cells, and it will actually accelerate the destruction of that C3bbb. Similarly, CD59, or protectin, is also on human cells to protect them. It will stop the MAC complex from forming, that MAC from forming. If you are deficient in DAF or protectin, CD59, do you know what disease you have?
Nocturnal hemoglobinuria.
Perfect. Paroxysmal nocturnal hemoglobinuria. PNH. If you don't have DAF or CD59. Very good. Okay. So another way this exact same system can get started is the lectin pathway. So lectin is just a protein that can bind to a carbohydrate. Period. In general, that's what a lectin does. Of course, mannose is a type of carbohydrate, so there's something called a mannose-binding lectin, or MBL, which is a lectin that binds to the carbohydrate mannose. And mannose is a carbohydrate that is found on pathogens and not on immune cells. And not on human cells. Okay, so this mannose-binding lectin can bind to something called MASP, or mannose-binding lectin-associated serine protease. Not something you need to know. Just know that MASP-binding lectin binds to MASP, which will then clip C3. And you make a bunch of C3b, and then things progress in the same way as it did before. So far, we've learned that C3 can spontaneously cleave to C3b, or MBL can bind to MASP, and then MASP can clip it. Okay. Complement can work in other ways, other than this traditional cascade of proteins. C3b can actually change into something called inactivated C3b, or IC3b. And when it does, it loses most of its function, but it's still able to grab onto pathogens, or what's called opsonize. It can opsonize pathogens. It grabs on to, pathogens, and it essentially decorates them and makes them more attractive to phagocytes. So when a pathogen gets covered with this IC3b, sign that the phagocyte should come along and destroy it, or eat that pathogen up. Another function of the complement system, moving back, if you remember when C3 and C5 are clipped, we talked about the C3b or C5b. Well, then there's that leftover part, that C5a or the C3a, which is sometimes referred to as anaphylatoxins. But they do a great job in attracting neutrophils and macrophages, which are phagocytes, to the site of infection where they need to be to actually perform their function. Okay. So the next part of the innate immune system we're going to talk about are the phagocytes, one of which is a macrophage. Right? If you think of the macrophage, you can think of this as a sentinel on the skin and other barrier tissue, other tissue that serves a barrier between the outside and your body. So these sentinels are just kind of sitting around waiting for something to happen. In the resting state, they express a few class 2 MHCs. These are the presenting proteins on the outside of cells. But then what can happen is they can get activated by signals from T cells or NK cells. That signal is interferon gamma. So interferon gamma is the first cytokine we're talking about. So don't get overwhelmed by all the different cytokines. Cytokines are just messages sent from one cell to another. That's it. So this one message we term interferon gamma. And T cells can secrete it, and they send it towards the macrophage, and that activates the macrophage, which then upregulates MHC2, and it begins to engulf those pathogens in the environment. Another option is that macrophages can actually get hyperactivated if it directly senses an invader. So this is done by pattern recognition receptors on the outside of the macrophage that recognize certain patterns. And patterns, these repetitive patterns, are often in pathogens and not so much in human cells. One of them that we'll hear a lot about this year is LPS. So LPS can bind directly to a pattern recognition receptor, like a toll-like receptor. And it will hyperactivate that macrophage. And when this happens, it secretes another cytokine called TNF, which we'll talk more about later. Like we talked about last week, though, macrophages can't do that by themselves. So they can call in help from another phagocyte that's in the blood circulation that can be made very, very quickly, which is a neutrophil. So neutrophil is very short-lived, but it's a very good phagocyte. It can be made very quickly in extremely large amounts. We know this because when we draw CBCs and someone's infected, we can see their neutrophil count go very high very quickly. When they're activated, they'll also produce TNF. And they'll go from the blood to the site of infection and do really the exact same thing that macrophages do. They just phagocytose these pathogens and destroy them. They eat them. But in order to get to the site of infection, there's a problem, right? They're not in the environment where the infection's going on. They're circulating in the blood. They actually have to get out of the blood vessel. And this is something we're going to learn about in more detail. But in order to do this, they use something called integrins and selectins to slow down as it's traveling in the blood through the blood vessel, and then stop or adhere to the side of the blood vessel and then squeeze through. Right, so this is done by integrins and selectins, like I said. So there's something called integrin ligands, and ICAM is an example of one that's found on the surface of vessels. There's something called selectin ligands on neutrophils. But when in an environment there is a sign of infection, like TNF, or IL-1 being secreted by the cells that are in the battle, or on the battlefield at that moment, then something will happen. It will cause the endothelium in that particular environment to express selectin. So then that selectin can bind to the selectin ligand on the neutrophil and can make it slow down. And then other things, other signals that are being secreted at the site of infection will cause neutrophils in the environment to express integrin. So not only will they slow down, Well, then they'll sense these things like the C5a, and then it'll express integrin, and then it will bind to that integrin ligand that is always expressed on the surface of cells or constitutively expressed on the outside of cells, and it will stop. Once it's stopped, it can squeeze through to endothelial cells to the environment. Remember, if this doesn't work correctly, you have LAD, leukocyte adhesion defect. It can't adhere to the side of the vessel and squeeze out. And then once it's out, it gets the rest of the way to the battlefield by sniffing out things like that C5a, which has come up a couple times, right? That byproduct of the complement pathway. But there's also something called fnet that is in bacteria and is unique to bacteria. And as those bacteria are getting phagocytosed or engulfed, the macrophages are spitting out fnet. And the neutrophils can actually sniff the fnet and follow it to the site of infection. All right, moving right along. Next cell of the innate immune system, NK cells. So NK cells are the only lymphocyte. That's, well, I'm not going to say that. NK cells are a lymphocyte in the innate immune system, right? So these are an example of a lymphocyte where the DNA does not rearrange. That cell is made, the DNA stays the same. They mature in the bone marrow and they proliferate in response to IL-2 and IL-15. You don't need to know that now, but I'll give you ways to remember that as we talk more throughout the year. And when they're activated, they are activated by interferon that can be secreted right by T cells on the battlefield, IL-12, or LPS. Remember, LPS is that pattern that can be found on pathogens. And then once it's activated, it can release inflammatory cytokines like more interferon and IL-2. So the job of the NK cells is that it will destroy cells by initiating apoptosis in that cell. So when would you want to do this? You'd want to do this when that cell is actually a tumor cell, or if it's virally infected, then you want that cell to die and not proliferate. Now when NK cells go up to other cells and check to see if it's abnormal in some way, if it seems to be a tumor cell, if it seems to be infected, it does this by a series, it looks at all the different receptors and it gets signals from a combination of activating receptors and inhibitory receptors. Depending on which set of receptors is more powerful at that moment, whichever has more expressed, NK cell is going to decide to either ignore that cell or to go ahead and initiate apoptosis or kill it. I found this to be a very tedious part of learning immunology, something to look forward to down the line, kind of getting a sense of which are activating receptors and which are inhibitory receptors. Okay. Any questions about the innate immune system? Okay, so the other big arm is the adaptive immune system. So cells in the adaptive immune system are B and T cells. They adapt by rearranging their DNA from the germline. They change. DNA is not the same in the completed cell. One of the benefits of this is they can actually become better at what they do, and when they do, they have memory. So we typically don't get infections more than once because you have memory and you can fight that infection off often before you become symptomatic. So B cells, so the next kind of bridge we call the B cell side of the adaptive immune system, the humoral immune system. B cells secrete antibody, which used to be thought as humors that got passed from person to person, or could potentially in a transfusion get passed from person to person. So the humoral immune system are B cells. So B cells are made in the bone marrow. And they have something on the outside, which is a B-cell receptor, which is essentially an IgM. And the IgM is made up of two heavy chains, which are these big, long guys, and two light chains. So then this part down here, well, actually, all of this gray is a constant region. This doesn't change. But then up here is a variable region where the DNA actually rearranges to become more specific for an antigen. And it does this by recombining three segments. So there's a V segment, a D segment, and a J segment. And the DJ segment rearranges first, and then the V rearranges onto the D and J. So when I was a fellow, I was taught to remember that DJs came before VJs. Disc jockeys came before video jockeys. To remember this little factoid, DJs or VJs. The DJ rearranges first, and then so you get a VDJ rearrangement. Then what happens is that this site here, both of these sites can bind to an antigen. This is the antigen binding site. And then this constant region dictates the function of the antibody. And right, I think that's all I'm going to say about that right now. Okay, so once that's happened, once that B-cell receptor or that IgM that is bound to the outside of a B-cell has rearranged, then that B-cell is called a naive B-cell because it has not been activated by an antigen yet. And I got this term confused many times when I first started reading this. I used to think, or I would get confused and think that naive means that it hasn't undergone rearrangement yet, but it's not. The B-cell's complete, done, but it's still naive. It hasn't been activated by an antigen. Okay, so when the B-cell is, this IgM is in the form of a B-cell receptor, right, so it's bound to the outside of a B-cell. These antibody binding regions will bind to an epitope of an antigen. Epitope is just part of the antigen that gets recognized by the antibody, so it binds to the epitope. If you look at this drawing, you can see that, okay, so here's the cell wall. There's not a big tail into the cell, so it actually can't send a signal very well, but it relies on two things called Ig alpha and Ig beta. And here, another names are CD79A and CD79B. Right, first example of more than one name. These can signal. So the antibody, the IgM, actually binds to the antigen, and then these Ig alpha and Ig beta do the signaling into the cell. And we'll see this over and over in the immune system that what does the signaling doesn't necessarily do the binding. The many molecules on the outside of the cell have to work together. Okay. So for the signal to occur, right, it needs both the Ig alpha, Ig beta, and the IgM. But then it can't just, it doesn't on its own generate a strong enough signal to get all the way to the nucleus. So one or two things have to happen to even send a signal. One is that there has to be cross-linking, and that if you think of the repetition on the outside of a bacteria cell, a bunch of IgMs all next to each other all have to bind, and then they all have to be signaling a weak signal to the nucleus. So that's called cross-linking, and that can generate enough signal to send a weak signal. The better option is that there's a second signal sent at the same time via a co-receptor. So co-receptor is a molecule that will actually bind to the same thing as the receptor does. In this particular case, that co-receptor is I don't know why that R's there. It should be D. CD what? CD21. So here's a nice picture where we have the IgM, and then you have your Ig alpha, Ig beta used for signaling, and then Here you have your CD21, which is a co-receptor that can amplify that signal and make sure it's strong enough to get all the way to the nucleus. To get activated, you need two signals. One, the antigen binding to the receptor, and you also need something called a co-stimulatory signal. So co-stimulator is a molecule on the outside of the cell that binds to something different from what the receptor binds to. The receptor binds to the antigen, so co-receptor also binds to the antigen. A co-stimulator binds to a whole nother molecule altogether. Okay, so for a B cell to get activated, it actually has to be presented something on another cell that has a co-stimulatory signal. In this case, it's something called CD40 ligand interaction. There's a CD40 binds to CD40 ligand. Another way that we exploit B cell activation is to use something called mitogens. So mitogens, like I mentioned last week, are proteins found in plants that force a cell into mitosis. If a cell is capable of dividing, it will force it to divide and get activated. This happens, though, in a B cell receptor independent manner. But we do this in the lab when we're checking to see if B cells work. We use mitogens to see if they'll activate. Okay, so once it gets activated, what's the first antibody that a B cell will secrete?
IgM.
IgM, very good. But then what can happen, remember that IgM has already undergone VDJ rearrangement. And it'll rearrange again once it gets a CD40 ligand signal, and it will change its constant region. It will splice out this M, and then it'll put in a constant region for a G, E, or A in the DNA. So then when that protein is eventually translated, then it can make a whole different protein. So it'll turn it into IgM, IgE, or IgA. So the first IgM is, when it's secreted, it quickly forms a pentamer. So there's 5 of them together. So it turns into a really big molecule that is very short-lived. Its half-life is about a day. If you made a bunch of IgM, you'd anticipate they would be all gone in 4 to 5 half-lives, 4 to 5 days. But it can also, it's very, very good at fixing complement by the classical pathway. So it starts the complement system. It's the third way to start the complement system. It basically does the same thing. It just cleaves at C3. An IgG can also fix complement. It's not as powerful as IgM, but it's pretty good. But there are certain subtypes of IgG that fix complement better than others. Right? The IgG subtypes are IgG1, IgG2, IgG3, IgG4. In the order in which they fix complement is 312. Area code in Chicago, 312. So IgM, and then IgG3, then IgG1, and then IgG2. All fix complement in that order. IgG, like complement, can also opsonize a pathogen for phagocytosis. It participates in something called ADCC, which we'll talk about more later. It crosses the placenta, and it's secreted in breast milk, often connected to a protein. And it has a very long half-life. That half-life is about 3 weeks. When we think about when we dose IVIG, we dose IVIG because we dose it, IVIG about once a month, once every 3 or 4 weeks. We like to do it every half-life. Medications that generally like to dose every half-life, and IgG is no exception. It has a nice long half-life, so we don't have to dose it that often. So an IgM will class switch to IgG in response to interferon. So if it senses interferon, the IgM is likely to become an IgG. Another antibody, IgA, is the most abundant antibody. It tends to guard mucosal surfaces. It's secreted as a dimer, so 2 of them together. So bigger than IgG, but not as big as an IgM, that pentamer. And that dimer is held together by a clip in the middle, which helps facilitate it across a wall for secretion, and it helps it become resistant to those GI enzymes, which are very, very good at, of course, breaking down proteins if you want it to, but IgA is quite resistant to that. IgA is also secreted in breast milk, not bound to anything. And IgM can class switch to IgA if it senses TGF-beta, cytokine TGF-beta. IgE, of course, causes mast cell degranulation, which is important in the defense against parasites, but we know IgE is better in this day and age. Because we don't have a lot of parasites around, we know it better as the allergy-causing antibody. So IgM will class switch to IgG in response to IL-4 and IL-5. Okay, so B cells also have one other unique property. They can undergo something called somatic hypermutation. So after they have rearranged, that receptor DNA can continue to mutate at a very high rate. And then the B cell undergoes some scrutiny, or that antibody undergoes scrutiny to see if that mutation has caused it to become more specific or less specific to the pathogen or the antigen. If it's less specific, then it dies out. If it's more specific, good, it'll be preserved, and then it'll mutate again and become more specific and more specific and more specific. So your antibodies are extremely, extremely specific for particular pathogens, which you want because you don't want antibodies to accidentally fight yourself, your own human cells. You want them to be quite specific to pathogens. So B cells, once they have, B cells can change into plasma cells. So they can go back to the bone marrow, and their sole job is to just secrete large amounts of antibody, and that's how they can fight infection. But then they also can be preserved as a memory B cell. And they're adaptive, so they have memory. The next time you have infection, that very specific antibody is already made and ready to go. Okay. Now, a bit about the T cell side of the adaptive immune system, which we talk about as the cell-mediated immunity. Humoral is adaptive, and then humoral B, cell-mediated T. All right, so T cells have to be presented an antigen on an antigen-presenting cell. And it does this via proteins called Class 1 and Class 2 MHC. So MHC type 1, I've got a picture of it here, is made up of one alpha chain and a beta-2 microglobulin, and I just noticed that I stole something that's not in English. It has a closed groove, so it only holds a very small amino acid, but that's okay, and we'll talk about this in a second, but that amino acid is packaged very carefully. It ends up being 8 to 11 amino acids in length, and it sits in this groove. And then can present to, of course, what type of T cell. CD4 or CD8. 8, very good. CD8. MHC class 1 by CD8. There are 3 different types of class 1 MHC, HLA-A, B, and C. And class 1 MHC is found in almost every cell in the body. It's true, that's not exactly true. What is true is that it's every nucleated cell in the body. So it wouldn't be found, for example, on red blood cells. But almost every cell in the body can present to a CD8 T cell because they'll have class 1 MHC. Not true of class 2, but we'll talk about that in a second. Okay, so class 1 is going to present proteins that are endogenous or inside the cell that has that class 1. And they may have gotten there because the pathogen has infected that cell. What happens is then the proteins, those pathogen proteins, get cut up in something called proteasomes, and then they're carried by a transporter across the endoplasmic reticulum. Remember what that transporter is called? CAP. Very good. It's carried by CAP across the endoplasmic reticulum, and then it gets loaded onto this class 1 MHC, and this whole package of class 1 MHC and protein is transported to the cell surface for presentation. In antigen-presenting cells, not all cells, there are things called LMP2, LMP7, and Macrophages, which can increase the amount of proteins that are suitable for presentation. So antigen-presenting cells still can more effectively present with MHC class 1, but all cells can present with MHC class 1. MHC class 2 is made of an alpha chain and a beta chain. Looks similar, though, to MHC class 1. I didn't put a picture up here. Has the same groove, but the amino acid doesn't have to be cut and chopped very nicely to fit into that. It can be anywhere from 13 to 25 amino acids long, so pretty big compared to what an MHC class 1 holds. So examples of class 2 MHC are HLA-DP, DQ, and DR. You find class 2 MHC only on antigen-presenting cells, not all cells, just antigen-presenting cells. So they tend to display exogenous proteins, for example, that have been phagocyte host by that antigen-presenting cell. So when class 2 MHC is made, it will initially bind to something to prevent any protein from binding in that cleft until it's almost ready to come to the outside of the cell. What that thing is that sits in there, prevents binding. Invariant chain. So that invariant chain prevents the protein from binding. And then when the time is right, when there's appropriate proteins to be loaded in there, there's something that will actually cause release of the invariant chain, which at this point, this is a minor detail, will actually be called a clip. The clip is taken out by something called HLA-DM, not to get confused with the MHC class 2 molecule itself. I hate this name because it is so similar to DR, DQ, right? Don't get tricked into thinking this is a class 2 MHC. It's not. It works to correctly make an MHC class 2 molecule, but it takes out that clip and lets an exogenous protein get in the cytoplasm of the cell, and then it goes to the surface to be displayed. This is a very cumbersome topic, so don't get weighted down on this yet. We're going to go into all this in a lot more detail and make it more understandable as the year goes on. Okay, so this MHC class 2, right, I said they're an antigen-presenting cell, so what are the antigen-presenting cells? Well, typically they're macrophages and dendritic cells, and B cells are the ones that are going to have that class 2 MHC and be presenting to T cells. And then just like in B cells, T cells are not going to get activated by just this MHC T-cell receptor interaction. It's going to also need a co-stimulator, and that co-stimulator comes with something called a B7 CD28 interaction. Don't get hung up on this either. Just trying to start introducing the concept that these activations happen with kind of a fail-safe. You take a safety off a gun before you can pull the trigger. It's kind of the same. You need two signals for this to happen safely: the co-stimulator and the antigen and receptor. Okay, so dendritic cells are also sentinels, just like macrophages are. They're the ones that initiate their immune system, so they get activated either by those pattern recognition receptors directly, excuse me, sensing something from the environment on the pathogen, or by TNF. But then its main job is not to just sit and eat the pathogens. It travels via the lymph to the lymph node, where it activates T cells to start the immune response. But it tends to be very short-lived. Macrophages we talked about before, right? Those are also the sentinels. They activated by interferon or pattern recognition receptors. And they can actually help by re-stimulating the T cell at the site of infection to let the T cell know that it needs to keep fighting. We will stop re-stimulating the T cell as that pathogen is getting cleared. And then, of course, B cells can act as antigen-presenting cells. And what it does is it actually binds the antigen to the B cell receptor, engulfs that whole complex, and then repackages it on MHC Class 2 and presents it to T cells. And they're very efficient at this. Okay, almost done. One quick mention of one more type of MHC. There's something we know about now, haven't known about for that long, called the Nonclassical MHC. An example of this is something called CD1, which is an MHC Class 1-like molecule, but instead of holding a protein, so far everything we've talked about is a protein getting held in MHC, well, it actually binds a lipid or a glycolipid. And then it presents to a T cell or an NK T cell, this glycolipid, but not a protein. This is the exception to the rule. We're going to talk way more about this. So the point here is really more that there's exceptions to every rule than to start memorizing. The fact that there's non-classical MHC is really meant to just kind of review what you've learned probably in medical school or college. Okay, so our review questions. Question 1, what DNA segments are the first to rearrange in adaptive immune cells?
TJ.
Very good, DJ, and then VDJ, and then VDJZ. Which antibody is the best at fixing complement?
IgG 312.
Yep, and even before IgG, what's the best?
M.
IgM, then IgG 312. Very good. And what molecule causes the release of the CLIP or the invariant chain in the MHC Class 2 presentation?
DM.
HLA-DM. Very good. Okay, that's it for today.